Hello everyone, welcome to the ITLS Academy. Today we are going to perform the experiment that is your media preparation. So before starting the lecture, we have to know that what is this culture media? Simply, a culture media is defined as any liquid or a gel-like substance which is used for the growth of the microorganisms like your bacteria and the fungi. So today the media that we are going to prepare is a nutrient agar media. So for the preparation of the nutrient agar media, we need some component. So first of all, we required a agar. Then we required a peptone, sodium chloride, and beef extract. If we have to prepare our media for 1 liter or 1000 ml, at that time the composition of this media, or you can say that the composition of these ingredients are as follow. Like we required agar around 15 gram, peptone 5 gram, beef extract 3 gram, and sodium chloride 5 gram. But today we are going to prepare it for 100 ml. So according to it, we are going to adjust the weight. But before preparing that media, we have to know that what is the role of all of these chemical. So basically, the first chemical, that is your agar, it is used as a solidifying agent. In a media preparation, the role of the agar is what? It is used as a solidifying agent. Then peptone is used to provide a nitrogen, which is very important for the growth of the microorganism. Then this beef extract, it is used as a source of many water soluble nutrients like your vitamins, carbohydrates, nitrogen compound, and different salt, which are very essential for the growth of the microorganism. And at last, we required a sodium chloride, which is used to maintain the osmotic pressure. So first of all, what we have to do, we measure a 50 ml of distilled water with the help of the measuring cylinder and put it into the conical flask. Now I am going to take a measurement of water. Now this is around 50 ml of the distilled water, which is used for the dissolution of all the ingredients. Then after that, I am going to add one by one all the chemicals into this conical flask. When all these chemicals are completely dissolved into this distilled water, then after that the remaining 50 ml water will be added. Now first of all, I am going to take a weight of agar. So I am going to switch on the weighing balance. You can see that it is 0. Now I am going to put a aluminum foil into it. When a weight are there, then what you have to do, you have to tear it. Okay, now it will be a 0. Now for the 100 ml of distilled water, the agar is required around 1.5 gram. So we have to take an exact weight. It is around 0.42. Now again I am going to add this agar into it. Now it is 1.20, but we required a 1.5. Again I am going to add. It is 1.34. We have to add until it reaches to 1.5. Now it is 1.6. Again, we have to reduce the amount of agar. Now it is 1.51. Now this agar 1.52 gram of agar will be dissolved into a distilled water. Now dissolve agar into distilled water. Then after that we have to add another ingredient, that is your peptone. But the amount of peptone is around only 0.5 gram because we are preparing it for 100 ml. Again, we switch on the weighing balance, put aluminum foil into it. Now tear the weight of the aluminum foil. Then after that, I am going to add a peptone. The weight of the peptone should be 0.5 gram. So we have to add it very carefully because weight is very less. 0.29, 0.44, 45, 49. It is now it is 0.51. Now I am going to add this into the distilled water. Now again we have to dissolve it into distilled water. Now the next ingredient is your NaCl. NaCl is also required 0.5 gram. Again we have to take a weight of that aluminum foil and tear it. After tearing, we have to add NaCl and it is around 0.3, 0.5 gram. 0.25, 44, it is 0.6, so we have to reduce it. That's why we have to add very carefully. Now it is 0.5. Now the sodium chloride is also added into this solution. Again we have to dissolve it for complete. Now the last ingredient is your beef extract. Now after taking the weight of the beef extract, that is 0.34, we have to add it into a conical flask in which all the ingredients are already present, and along with it 50 ml of distilled water are also present. Now we have to mix it properly so that all the ingredients are completely dissolved into it. See, it is completely dissolved. Now after that we have to add the remaining 50 ml of distilled water into it. Now I am again to measure it with the help of the measuring cylinder. Now this 50 ml of distilled water are again added into the media. Okay, one thing that we have to take care that we don't prepare the media like from this level. We have to prepare it till this line so that when we autoclave it, it will not be contaminated. Okay, now after that, when all the ingredients are dissolved into the distilled water, we have to uh, measure its pH. Okay, when we are measuring its pH, it's come around 6.8, but we have to adjust its pH to 7.4 plus minus 0.2. If we are not adjusting its pH, it will maybe it does that agar is not solidified. So the maintaining of the pH before sterilization, you don't have maintain the pH after sterilization because it will create a contamination. You have to maintain its pH before autoclaving. So now I am going to maintain its pH, and you know that when you are maintaining the pH, you have to calibrate your pH meter. So our pH meter are already calibrated. As you see that it is already calibrated, and how we calibrate our pH meter, the video related to this is already uploaded in our account. You can go and check it. Now you can see that this is our media and this is an electrode. So first of all, we wash this electrode which are already dipped into the KCl with the help of the distilled water. Now after that, we pour this electrode inside the media. Now I am moving the function key from standby to the pH. See that it is come around 6.444 or 48. That means that we have to add a NaOH solution into it so that it reaches to 7.4. Now with the help of these micropipettes and the tips, I am going to add a NaOH into it. You have to add NaOH very slowly, otherwise its pH will raise. So I am going to add 2 to 3 drop of NaOH into it and mix it properly. After mixing it, I am going to take a pH with the help of the electrode. It is around 6.1. So that means that we have to add more NaOH into it. Again, after adding NaOH and proper mixing, again we have to take a pH. You can see that the reading is around 7.4. That means that we have to add more NaOH into it. It is 7.02. Again, we have to add a NaOH into it. Again, its pH is not reached 7.4, so we have to add more NaOH into it. Again, with the help of proper mixing, we have to mix the NaOH into it. Again, we are adding it. It reaches 7.4. That means that our pH is maintained. Now again, we have to wash it with the help of the distilled water and dip it into the electrode. Now this is how we maintain our pH with the help of that digital pH meter. Now our media is ready. Now what we have to do, we put a cotton plug into it and wrap with the autoclaving paper and autoclave it at 121 degrees Celsius, 15 psi pressure for 20 minutes inside the autoclave. Once it autoclave, then it is ready for a pouring. After that, we perform our next experiment, that is your pouring or spreading with the help of these media. So this is all about the media preparation. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also you can subscribe our channel on YouTube and get our app from a Google Play, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.